Yeah, good morning, everybody. Thanks for coming. So I'd like to begin with something that inspires me since I was a student, so 10, 20 years ago. And it looks like a thing of the future, but what you're seeing here is actually over 1 billion years old. So what you're seeing here is a artistic illustration of a very important enzyme. It's a molecular machine. Every living cell on Earth has it, because every living cell on Earth needs ATP. That's the fuel on which cells run. And this enzyme actually makes the ATP. You find it in the mitochondria, in the cells of your body, in the membrane. And when you eat and breathe, actually, inside of these mitochondria, there's an ion gradient created. And that ion gradient, meaning there's a higher concentration of ions in the top part of this picture, and there's a lower concentration of ions in the bottom part of this picture. And what the ATP synthase does is actually rotating, so driven by an ion current, as you can see here, and the rotation drives cyclical deformations in this mushroom-shaped part of this enzyme. And driven by these rotations, actually, the adducts of ATP, adenosine diphosphate and phosphate molecules, they're being basically pressed together in this kind of mushroom cup, and out comes a higher energy molecule, ATP, adenosine triphosphate. These machines, they're about 50 nanometers small, so it's smaller than the latest generation transistors, for example, which are used in computer chips, and then there are about 50,000 atoms, which are precisely arranged in the structure of this molecular machine. And they're quite amazing. If you look at the data sheet, they can do up to 100,000 revolutions per minute, noticeable torque, and they can make 3 ATP molecules per second. So it turns out that our entire body weight is actually being used up for the entire day in sheer mass of ATP. And only 10 grams of ATP synthases in your body make all these, tens of kilograms of ATP every day. So these are, I believe, amazing molecular machines, and it shows us what could be possible if we knew how to build such a thing. So today, if we look at modern-day synthetic biology or biotechnology, what we're doing essentially is some type of bioprospecting. We can look into nature and look for function, and then take out some molecules that we deem interesting, and we tinker and repurpose them. Famous examples are antibodies, for example. There was a very convalescent patient, for example, who survived Ebola. The blood has been examined, and antibodies have been identified, and then taken out of the blood and produced on large scale, and this is now the only existing therapeutics against Ebola, for example. Other famous examples are DNA polymerases. These are molecular machines that can copy DNA. They're being used around the world as we speak in mass quantities to make PCR tests for COVID. So this is an example how basically such kind of machines that you already have in nature, they were developed in the course of evolution, how they can be used for technology. However, what if you want to do something which is very unlikely to find in nature? Then you have a problem. You have a need, you can specify it, but in order to satisfy the need, you would need to be able to build a device or molecular machine that can satisfy this need, right? So you need to be able to build from scratch. And this is the problem that I've been working on for now over the decade, so I've been interested in. And we have been working on a particular nanofabrication technique that you can call sequence programmable DNA self-assembly. It's also known as DNA origami. And I want to briefly give you a high-level description how that works. So basically what we're doing is we're sitting in our offices, and we have a rational design process that allows us to come up with sequences for hundreds of DNA molecules, DNA single strands. And they're designed in such a way when these molecules are actually made and they're mixed together in one test tube, in one solution, they will self-assemble and fold by forming base pairs into a three-dimensional structure which is already fully encoded in the sequences. So we don't need to put these molecules together, they find the right spot themselves. Here's an illustration of this process. It's not meant to be a physically accurate illustration. It's more like giving a gist of how the process works. So typically we have 2 main components. We have a very long DNA single strand, which we call a scaffold molecule. And then we have many short DNA molecules, which have designed sequences, which we call staples. And then so we mix them all together, and then through the base pairing interactions, which are encoded in the sequences of these molecules, they will find themselves buffeted by Brownian motion solution, and they will fold up this long scaffold molecule into a structure, and which in this case happens to be a 40 nanometer long rod consisting of 10 DNA double helices, which are bundled together. So and the beauty of this process is that you can plan ahead where in space, inside of this nanostructure, certain DNA bases will be. And every DNA base here, this one contains about 1,000 maybe, can be individually addressed with a chemical modification. So that's a quite unique nanofabrication method. However, this was fiction, this was an illustration. Now I want to show you some reality. So we're seeing here everything that's yellow and blue. These are structures that we actually made, and they consist 100% of designed DNA molecules. And these aren't models or computer models, so what you see is actually data. These are structures that we solved experimentally using cryo-electron microscopy. And to give you a sense of scale, I also included 2 objects of natural origins on the top row. You can see a hepatitis B virus core capsid, which is a 35 nanometer object. This is a spherical ball in the top. And then the second row from the top, you see a slightly smaller object, also in magenta. So that's a membrane protein. I'll run it again, actually. Well, I don't. That has a molecular weight of 800 kilodalton. So the main point I'm trying to make is that DNA nanotechnology has matured to the point that it can fabricate almost any kind of shape. You can experimentally validate them and potentially refine them. And these shapes that you're making, they're commensurate to the dimensions of natural macromolecular assemblies that you can find in a cell. So let's take a closer look at one of these objects to give you a better sense how they actually look like. So we can zoom in, and with the data we are able to collect with the cryo-electron microscopes, we can actually see individual helices with all the sort of beauty. We can see the major minor grooves of DNA double helices, but this is a quite unusual shape here, right? Because all these molecules are kind of put together into some kind of cubic object. So this object doesn't have a particular function. It's meant to be more like a sculpture, which we use to think about some design rules. So then having that kind of data, we can also fit atomic coordinates in these kind of electrodensity maps. And then we basically have a list where we can really say, okay, here's a base and has these coordinates in space, and you can quantify and look at some of these geometrical properties. And now what you're seeing here on the left-hand side is the atomic model basically derived from the structure I just showed you. On the right-hand side, there's another atomic model, and it happens to be the version 2 of the same object. And version 1 was slightly twisted, had some defects, and then we went and modified the design, and we made a straight version. So this basically to illustrate that you can do something which is known for macroscale engineering, iterative processes of design refinement. You can also do this on a nanometer scale using DNA fabrication. So over the years, we cleared, I believe, many of the roadblocks that stood in the way of using this type of nanofabrication for actually some real-world applications. And today, I want to use this opportunity to talk about 2 devices, well, 1 device and an actual machine that we made using DNA nanotechnology, where we actually specify the need and we build from scratch. So the first thing I want to talk about is these virus straps, which basically is not a machine. It's really something that's just, it can do one task, but doesn't, perform any work or something like that. So the virus straps. So first, let's talk about the need. Well, there's a huge societal need here. So hundreds of millions of people every year are affected by over 200 non-viral diseases. Well, our mind is focused on COVID maybe, but there are many other viral diseases out there, and very few of them actually can be treated. The common route is prevention by vaccines, but an actual treatment of an acute infection almost hardly exists. So in current antivirals, the few that exist, they target really only very specific viruses, and something that we know from antibiotics, like a broad-spectrum antibiotics, it simply doesn't exist for antivirals. So this is a societal need, and we're thinking, is there some kind of universal way to prevent viruses from spreading? Well, we all know one, which is quarantine, but we don't want to put humans in quarantine all the time. So here's a wild idea. What about we just put viruses in quarantine? So all viruses, they need to interact with cells, because they need the cells to replicate. So they need, at some point, they need to interact with cell surface and invade it. So what if we just built some kind of macromolecular cage, like some kind of jail for a virus? So we build this using nanofabrication, and then we coat it from the inside with some molecules which have specific affinity for the virus we want to trap. And then the idea is that sort of these cages, they will circulate through the body and trap the viruses inside of the cage, much like flies are being caught on fly paper, and when they're inside of a cage, the viruses can no longer interact with cells, and the infection spreading is contained. So to put this wild idea, admittedly, into practice, we engineered something what we call a programmable icosahedral shell canvas. So these are DNA building blocks. So DNA is used as a construction material again. We make these kind of triangular building blocks which are capable of assembling into higher order shells. So you see some of these examples, and the assembly process needed to work so well that we can actually use it, right, in the real world. So we were able to solve structures of the individual shell prototypes that we made. And here's a photograph of a solution containing many copies of one of these icosahedral shell prototypes in solution. You see, it works quite well. They actually even look a little bit like viruses. So now, in order to trap viruses, we needed slightly different shells. So we learn how to stop this kind of fabrication process so that it forms shells that have defined apertures. So, for example, we can make a half octahedron. We can make a half icosahedron, or maybe a 3/4 of an icosahedron. And again, these are cryo-electron microscopy structures, meaning that, and if you can solve a structure, it means it is a well-defined object in solution. If you cannot solve a structure if it's some kind of goo. So then what we do is we coat inside of these half icosahedra, for example, and then we can go and trap actual viruses. So here's a cryo-EM structure where you see a chikungunya virus particle, a tropical disease, actually trapped in such a DNA shell. And again, this is an experimentally validated structure. You see the virus sitting inside of the shell. The shell is 85 nanometer in diameter. And I mean, I've been showing this slide for a couple of times now, and every time I show it, I get so excited because it's just this whole notion you can build something by a rational design process which can actually grab an entire virus and engulf it. To me, that's very satisfying. So then here's a photograph where you see many chikungunya virus particles, and all of them are fully encapsulated by these kind of DNA shells. So again, that this actually works. You also see some triangles and fundamental building blocks lying around, which are from these kind of shells. So it works so well that we actually were able to trap a whole zoo of viruses. So we use different types of shells, different diameters, to trap a whole zoo of different virus types. So on everything here, all these images that I show you here, they have the same scale. So on the top left, we can start this adeno-associated virus. Viruses trapped in these half octahedra. There's polioviruses, dengue viruses, noroviruses, human papilloma virus, SARS-CoV-2, chikungunya, which I just mentioned, rubella in the bottom left, adenovirus, and then the latest entry here on the right are influenza viruses. So influenza viruses, as the latest development, they were a tough target because influenza viruses are not only bigger than all the previous viruses that we were able to attack, they're also pleomorphic. That's the term from biology, meaning they have quite heterogeneous shapes. So we managed to make a new type of shell which can cope with this kind of shape heterogeneity and still faithfully engulfs the enteroviruses. So all the viruses on the left-hand side, they are trapped with the same coating even. So this whole idea of making a broad-spectrum antiviral, I think it becomes tangible. Okay, so then we're going too much in further detail. We were able to show with live human cells and cell culture in vitro, as we call it, that when we trap the viruses, they no longer infect cells. We can really stop this infection process. We've shown this with multiple virus model systems, and now we have moved on and have tested already whether the virus trap, these cages, whether they actually safe to use in animal models. So there haven't been any adverse effects so far. So that's also very nice. De-risks the process quite substantially. And now what we're doing now is we're doing in vivo efficacy tests where we're trying to identify how and under which circumstances and under which conditions we can apply these cages so that we can actually contain an infection or even prevent an infection in mouse models. So the vision is that maybe one day we'll have some kind of programmable broad-spectrum antiviral. Maybe we can use it. We need to figure this out, whether it will work, where we can actually use it to treat acute infections. It will certainly very likely work as a pre-exposure prophylaxis. So here the idea is that, you, for example, administer these kind of cages to people who are at risk, medical personnel, or elderly people, and critical personnel. You can administer travelers, or maybe even to everyone in the world. If you're going out and you want to protect yourself, you get a dose of the virus cages, which will take care of you. So that's the vision. So and then thankfully some people share this vision. So this project, even though it's kind of wild, admittedly, we got funding from the European Union first and formed a consortium to put this into practice. And then I'm very thankful that also the Federal Agency for Disruptive Innovation saw some value in this approach, also supporting this. And we have now formed a company, Capsutech, in which we're trying to bring this virus trap as a therapeutic to the market. Then I also want to highlight the people behind this behind this work. So the two actually present in the room, so we can reveal yourself. Wave, there's Christian, and also Alba. They're working really hard to advance this project. Okay, so now I have some time left. I can talk about a very recent development, nanomotors. So again, I want to specify the need. And this is a very high-level need. So we know motors have powered the industrial evolution. You can't think modern-day technology without motors, an integral part of everything. We do not yet have nanoscale motors, but we know, we're certain, that nanomotors could do amazing things. Think of the ATP synthase that I showed you. Maybe you can use nanomotors to make chemical synthesis in a totally different way than we do it today, just as one example. So this is the need. Can we make artificial nanomotors? And today, very happy that I can actually introduce the world's first electrical motor that was made from DNA. So this was actually published yesterday in the journal Nature, which for scientists, academics like me, is a really big deal. For people outside the real world, maybe not, but it means it's kind of significant development. So the world's first electrical motor made of DNA. So again, before I start talking about it, I want to show the people behind their work. So the top row are all the students and fellow scientists in my laboratory, which have made this a practice. And Anna Katarina was really like the tenacious person who worked for 6 years on this project to put it in practice. And we had some help from Enzo Koppberger and Fritz Simmel, the partner lab at TUM, and also from Jonas Isensee and Ramin Golestanian from Max Planck Institute, Dynamics and Self-Organization in Göttingen. So now, in brief, how does a motor look like? Well, the motor has 3 components. It has some kind of pedestal with which you can fix it on a surface. And the pedestal is really small, 30 nanometer, 40 nanometer high. So it's about the same size as a modern day transistor, for example. And then there's a triangular platform, which is colored in blue, which will be fixed onto the pedestal. And then we have a rotor arm colored in orange, which you can fix on top on a pivot point. So then what we can do is to drive the rotation is we just need to apply an alternating current electric field. That's the only thing you need to do. Just stick 2 electrodes in solution, have an AC field on, and the motor starts moving in one direction. So this is how it looks like in an electron microscope. You see this long lever arm. It's actually a triangular platform on the left. So now I want to show you some motors in action. So on the left-hand side, you can see a region of interest from of a movie that we recorded in a very sensitive fluorescence microscope. Each pixel is a 100 nanometer, and we see the diffraction limited spot there, which is coming from the photons, the light emitted from some fluorophores, which are positioned at the tip of this rotor arm. So now I'm going to run the movie. So we're tracking the tip of the rotor arm. Basically, we're tracking the orientations. And so initially, we don't have an electric field on. So the motor just does random Brownian motion. So this is what you expect. And then we turn on the electric field, and it starts rotating. So now for you, that may be not so easy to recognize, but it actually rotates in a counterclockwise fashion, very possessively, and with a nice speed, about 250 RPM in this one. So and just to show that this actually rotating counterclockwise, I have a slowed down movie of the same motor, just one section. You can focus on the rotor arm tip. So there's some stochasticity to the movement, but it actually really moves processively counterclockwise. Now, interestingly, we can also control rotation of direction. I'm just not going to explain it how it works, but I'm going to show you how that works. So here's another motor which we run counterclockwise. Again, initially the field is off, and then we turn it on, and then it starts changing its behavior and moves in a clockwise fashion. Again, this is hard to see in this video, but again, there's a slowed down section of the same movie. You can see basically you can track the tip. Again, it takes occasional backstep, but most of the time it moves forward. So that's an actual motor. So now what we're seeing is sustained rotation of viscous medium. That's like swimming. You do some work, and all the work you do is dissipated in the water, in the friction, right? The question is, though, can these motors do actual additional mechanical work? Can we, for example, make them wind a spindle, a molecular spindle? So we made a new variant of the same motor in which we included a torsion spring at the pivot point. So now if the rotor arm rotates, it will actually wind the spring. So it's like you wind the torsion spring if you have a mechanical clock, for example, and you turn the crown. So and it actually worked. So I'm showing you some exemplary traces, again, from single particles, single motors. On the top left, you see a motor At first the field is off, so it's the green phase, it does some random motion. Then we turn on the field, and it goes counterclockwise, upwards actually, and then we turn off the field, and then it immediately changes the rotation direction and goes down again, which means it had some internal stored energy that made it go processively into the other direction. So that's the stored mechanical energy in these springs. And then the other graphs are basically examples, repetitions of the same kind of experiment. So here's a motor data sheet. So we made an artificial molecular motor. It's the best performing motor so far. It's up to 250 RPM speed, and it's totally limited by the friction of this big lever arm. The solution can do up to 10 piconewton nanometer torque. That's minuscule. That's 10 to the power of 10 minus 21 Newton meter. So that's a very small number, but it's not far from what the ADP synthase actually can deliver. The power output is about 250 piconewton nanometer per second. So that corresponds to 250 times 10 to the power of minus 21 watts. It's again a very small number, but if you think about the weight of this machine, it's 2.5 times 10 to the minus 20 kilogram. It's about 10 watts per kilogram if you extrapolate it, which is notable, I think. So then what's next? Maybe what we can do with this? Well, an interesting perspective is that whether we can somehow make a new prototype where we can use a chemical fuel to power rotation, or invert the process. Now that we know we can drive a motor, can we use the rotation of such a motor to drive some kind of uphill chemical synthesis, much like ADP synthesis does it. And if we manage to do that, we can have these tiny little chemical reactors, we can put them on a substrate maybe, and they will make a chemical compound of your desire, and the only thing you need to do is apply a little bit of AC voltage. That's at least a naive vision that I have that could be maybe take some place in the future. At the end of my talk, I showed you two devices that we made, one device and one machine, by a rational design process, a new type of nanofabrication. All this work would not have been possible without the financial support from many sources over many years, and the patience that they had in supporting us, the German Research Society, European Research Council, European Commission, and lately also the Federal Agency of Disruptive Innovations. I'm very thankful for all their financial support we have received. And last but not least, I want to thank all the nano engineers who have worked and built up the technology over the last years in my laboratory. About 60 people now. Yeah, without them, none of this would exist. So I'm very thankful. Yeah, thank you for your attention.
Thank you so much, Hendrik, for this great opening. Well, stay on stage, because I already asked Sprint, who comes after you, if it's okay if you make 1 or 2 minutes longer, and you made it in time, so we can take one question from the audience. Sorry, I'm messing things up, but I know that they are relaxed and they are not. All right, if not, just grab him. He will be here. Oh, there's a question in the back, right in the back. Some short light.
Yeah, Sean,
he gets a mic. Could you please repeat?
The question was shedding some light on manufacturing process. So basically, you make these molecules, for example, by chemical synthesis, or you can use cells, E. coli cells, for example, to make the DNA molecules, and then basically you have a big pot. You just dump everything in, you stir, and heat it a little bit, and then you have that's it, essentially.
So that was quite easy. Thank you so much, Hendrik Dietz, for this fascinating opening keynote. Thanks. Warm applause.